Welcome to the next rendition of our Nano Explorations seminar. I'm glad that you can join us after the Memorial Day weekend. I hope you had a relaxing time with your family or friends. The talk today is going to be delivered by Daniela Espinosa. She will give us a very inspiring talk on different ways of thinking about how to engineer myelination in vitro. And she will tell you more about it. Daniela Espinosa-Hoyos works with Professor Kristen Van Vliet, Professor Dan Anderson, and indeed is considering graduating later this year in August, so that she can tell you right now a variety of achievements she had indeed experienced over her time as a graduate student. So Daniela, please take over.
All right, so hello everyone, good morning. I'm going to, I'm happy to be here today to talk to you about my PhD research in the lab of Professor Kristen Van Vliet in material science and biological engineering. So my work is motivated by the prevalence of myelin diseases in the central nervous system, which consists of the brain and the spinal cord. Multiple sclerosis is one of the most common one of these diseases affecting millions of people worldwide. And despite extensive efforts, we still don't have a cure for MS or any of the myelin diseases. The loss of neurological function in multiple sclerosis stems from the progressive degeneration of neurons. Neuronal axons are covered in a protective membrane called myelin, shown here in green, which allows for the fast conduction of signals necessary for cognitive and motor skills. Myelin is generated in the central nervous system and maintained by oligodendrocyte cells, these OLs. Other glial cells called astrocytes and microglia are also important for brain homeostasis. MS usually begins with an immune-mediated inflammatory response. Immune cells are activated and cross the blood-brain barrier, recognizing myelin as a foreign agent and attacking it. Astrocytes and microglia are activated and they provide both neurotoxic and neuroprotective support. This immune-mediated inflammatory response leads to myelin loss, axon degeneration, and destruction of these glial cells. While some recruited oligodendrocytes can remyelinate these axons, most of them become apoptotic or they die, or they become dysfunctional. The drug development pipeline for neuroprotective therapies consists of years of discovery and development, where cells, small molecules, and biologics are first tested, and top candidates are further developed in a range of in vitro preclinical models. The top candidates go on to in vivo experiments with animal models, and with many more years and some luck, you might get a candidate that makes it to clinical trials, where most therapies in the neuroscience space end up failing. This paucity of discovery and translation has forced the community to really reimagine the drug development process and think about how we tackle this complex disease. This includes innovating on more predictive or translatable preclinical models. One overlooked aspect of myelination is that the myelinating cells, these oligodendrocytes, are mechanosensitive. That is, the various cell types along the oligodendrocyte lineage can sense the physical and mechanical properties of their environment. And this physical environment is continuously changing throughout development, aging, and disease. During my thesis, I have further discovered that the human oligodendrocytes are also mechanosensitive, and that different patient cells may respond differently to the same mechanical cue. So this now adds a new layer of complexity that may be important to consider in the development of therapies for diseases like MS that are already profoundly heterogeneous. Despite this mounting evidence of oligodendrocyte mechanosensitivity, oligodendrocyte biology is generally studied in the dish using materials that poorly represent the physical and mechanical properties of brain tissues and cells. It was first observed in the 1990s that oligodendrocytes can wrap myelin-like membranes around inert three-dimensional objects in the absence of neuronal signaling. And this idea that myelination can proceed in the absence of neuronal signaling presented an opportunity to bridge the trade-off between biological accuracy and engineered reproducibility and throughput of current preclinical models. So several systems, as you see here, have since then been refined using glass and polymer fibers to capture some properties of biological axons. While some of these capture essential features of the myelinated oligodendrocyte axon unit, they have several key deficiencies. Primarily, they sacrifice true three-dimensionality for the sake of throughput, as is the example of these cones, and the mechanical properties of the materials remain orders of magnitude higher than those of brain tissues and cells. So we conceptualize these artificial axons. These reductionist microstructures would capture some of the key physical and mechanical properties of neuronal axons. Namely, they're extremely thin, around 1 micron or lower in the brain. They're extremely compliant, around 1 kilopascal Young's modulus. They're relatively uniform and cylindrical, and they're closely packed into bundles. Furthermore, they're unsupported for spans that can accommodate three-dimensional wrapping of physiological lengths of myelin internodes. And finally, for this to be useful, myelination needs to be quantifiable, and the device or assays developed with it need to be compatible with cell culture and drug development pipelines. So the two different orientations you see here, horizontal or vertical, are meant to facilitate faster acquisition of certain readouts. For example, segment length from the horizontal orientation, or concentric wrapping from the vertical orientation. While the basic architecture of these structures was simple enough, the combination of the design parameters posed a great challenge for fabrication and could not be achieved by conventional stamping, molding, or patterning methods. So we considered several additive manufacturing and microfabrication technologies, and chose projection microstereolithography. This technique, refined by the Fang group in mechanical engineering here at MIT, features additive maskless photopolymerization of microstructures with good compromise between throughput, resolution, spatial control, and flexibility of fabrication materials. Briefly, 3D CAD models are sliced, and then these digital masks are sent to a projector equipped with a micromirror device that then shines the mask onto a bath of photocurable resin through a series of reduction lenses. And an XYZ stage is used to shift the printing plane to build on the size of the 3D structures. The practical capabilities of the technology are ultimately determined by the fabrication material or resin. And these resins typically consist of one or more monomers, a photoinitiator to initiate a free radical polymerization, and UV absorbers to control the vertical resolution, and a solvent. So this endeavor required the development of new resins and materials to achieve the often competing properties of biocompatibility, low mechanical stiffness, and manufacturability. In fact, the materials typically used in oligodendrocyte cell culture, shown here on the right, including previously developed axon-free models, are stiff, but they're easily manufacturable. On the other hand, compliant and biocompatible materials, shown here on the left, are closer to what the brain is like, pose great challenges for microfabrication, the length scales and mechanical properties that we are targeting. So what I did was develop hybrid hydrogels formulated with hydrophilic and high molecular weight polyethylene glycol precursors commonly used in biological applications, and hydrophobic and short acrylates commonly used in microfabrication for their efficient polymerization kinetics. So I copolymerized hexane diol diacrylate, or HDDA, and tetrapeg acrylate, or STARPEG, to form these hybrid polymer networks with hydrophobic and hydrophilic properties. STARPEG imparts mechanical robustness as well as stability in solution relative to high molecular weights linear pegs, and HDDA acts as a cross-linker, providing hydrophobicity that helps reduce or control swelling and hydrolytic degradation of the network. This hybrid hydrogel offers oligodendrocyte cell viability or biocompatibility comparable to that of plastic, which is standard, and it's significantly improved over the virtually zero viability that we found HGDA had. The swelling capacity of the material can be modulated, and it can also be manufactured 3D printed with comparable resolution and speed as HGDA using projection microstereolithography. This hybrid material can also be tuned by changing its chemistry and microfabricated to span the range of mechanical properties of brain tissue, as well as other soft tissues of interest. Overall, this material, which we call HSP for HGDA STARPEC, overcomes the limitation of the single monomer systems of HGDA and STARPEC for manufacturing of our artificial axon designs. So we optimize the materials and the methodologies presented here to fabricate these artificial axons. These are examples of the vertical orientation of those pillars that I showed you before. The spacing in between the pillars is small enough to facilitate the engagement of multiple artificial axons by a single cell. And the regularity of the structures was also crucial to facilitate automated imaging and image analysis. The artificial axons fabricated with these materials can be functionalized to facilitate migration of oligodendrocytes, engagement, and wrapping of myelin-like membranes, which you can see here in green. And these are the horizontally oriented artificial axons. Importantly, we have also tested the compatibility of these assays with common drug development practices in pharma, including the use of multi-well plate formats, high throughput confocal imaging, and semi-automated image analysis. I further use stem cells to make human oligodendrocytes. So everything you saw before was using rat cells, primary rat cells derived from brains. But now we can make human oligodendrocytes from cells derived from different patients or healthy donors to establish these human myelination assays, where now we can, for the first time, reproducibly visualize and quantify human-like myelin wrapping in the absence of neuronal signaling. So we can now use the simplicity, the control, and the reproducibility afforded by this system to, for example, further study the heterogeneity of oligodendrocytes from multiple sclerosis patients, and study the crosstalk between immune cells and oligodendrocytes in the context of myelination, but in the absence of neuronal signaling. So in vivo, this is what myelin looks like. The myelin sheath this is a cross section. It's compact. It consists of periodically wrapped layers, about 10 nanometers thick, of fused intracellular oligodendrocyte plasma membrane. And this ultrastructure is usually resolved with transmission electron microscopy, which is what you see here. So we have so far demonstrated that our samples can be processed by standard methods used for scanning and transmission electron microscopy. Imaging, unfortunately, has been paused due to COVID-19, but thus far we have at least a few images where we can see evidence of artificial axon wrapping, which is reminiscent of our typical observations from confocal microscopy, which is what you see here. But it still remains to be determined whether we have this type of compact myelin in our system. And this is basically the standard in the field to determine whether myelin is functional or not. An important feature of this platform is that we can image live or fixed samples and readily quantify true three-dimensional wrapping using conventional fluorescence microscopy. Therefore, the readouts you get in the system are not the typical two-dimensional readouts obtained in other traditional models where wrapping is typically assumed, but it's not confirmed. So I showed that not only can we quantify wrapping, but we can also recapitulate physiological features of myelinated axons, such as the distribution of the sheath lengths. And we can modulate these properties by changing biochemical or physical properties of these fibers, in this case, the coating of the fiber, which helps the cells attach and interact with them. And furthermore, I found that indeed the two-dimensional readouts previously used, namely the alignment of proteins that correspond to myelin, may mask or distort the results of making these comparisons. We can also readily manipulate the physical properties of the artificial axons to study their impact on oligodendrocyte biology. And this now allows us to look at oligodendrocyte mechanosensitivity in 3 dimensions. Here, we vary 2 properties independently, fiber stiffness and diameter. And we found that both stiffness and diameter impact the extent of oligodendrocyte engagement and wrapping with the fibers. Importantly, this is the first evidence that mechanical stiffness not only affects oligodendrocyte differentiation or maturation in 2 dimensions, as previously shown, but that it may also impact the 3-dimensional process of myelination. Under the particular conditions of this study, stiffer fibers of a certain diameter were more readily wrapped in MBP positive or myelin basic protein rich membranes than compliant fibers of the same diameter. So now we have a biomimetic platform compatible with human cells that reproducibly recapitulates important biology and allows us to pursue basic scientific questions. And we are pursuing several different research directions. One of these is improving the resolution and the throughput of fabricating these artificial axons, which will be crucial if we want to implement this platform in drug development pipelines for things like high throughput drug screening. So layer by layer fabrication of these hydrogel microstructures can be time consuming. We hypothesized that instead we could take advantage of the optical properties of the HSP material developed here to self-propagate hydrogel pillars using a single light exposure. This phenomenon is known by many names, including self-propagated polymer waveguides or self-written polymer fiber prototyping. It is basically based on nonlinear optical properties of the materials, specifically intensity dependent refractive index. So as the resin is exposed to UV light through the photomask, the material begins to polymerize. This causes a change in the refractive index, the local refractive index, relative to that of the surrounding medium. This traps light and focuses it. This process continues and intensifies, forming these polymer fibers until reaching some saturation as the polymerization reaches completion. So various experimental and parameters and material properties can be tuned to intensify this light focusing and extend the propagation of the fibers. To allow for the self-propagation of HSP pillars in the Z direction with a single exposure, we increase the light penetration depth and slow down the polymerization kinetics by changing some experimental parameters. And we discovered that photopolymerization of HSP pillars of aspect ratios of up to at least 1:75 could be achieved with a single exposure using the same projection microstereolithography system. This was reproducible with materials of Young's moduli spanning approximately 0.1 to 140 kilopascals. Notably, we could achieve fibers, as you see here, with diameters beyond the theoretical resolution of the optical system imposed by the optical system, which was about 1.36 microns per pixel. So this represented a significant improvement over the lateral resolution previously achieved using the layer by layer fabrication approach. The single step process also significantly improved the throughput or the fabrication speed, circumventing the time costs associated with stepwise vertical translation of the platform. This simplicity and uniformity of the vertically oriented artificial axons accomplished with a single exposure motivated us then to pursue more simplified fabrication methods, which then could be amenable to large area fabrication to generate a large number of samples in less time for things like high throughput drug screening, where you typically require multi-well plates full of these structures. So specifically, we reproduced the self-writing process, but this time using a white light source transmitted through a physical photomask, a fairly simple process. And this represented, as expected, a significant increase in throughput, circumventing now the additional time cost associated with horizontal translation in the projection microstereolithography system. And the freeform method was also compatible with in-well fabrication of artificial axons. So we can now fabricate them inside the wells of micro well plates, which are typically used for biological assays. And further optimization is still necessary. This is also work that was halted by COVID. So we need to further optimize process parameters to improve the diameter and the shape of the pillars, but so far it seems quite reproducible. So these advancements allowed us thus far to run experiments intended to determine whether the platform could in fact be used for drug discovery and development of uniquely human diseases of the central nervous system. Specifically, I demonstrated that we could stimulate myelin-like wrapping using various repurposed drugs, and that this stimulation of wrapping was dose-dependent. Therefore, this preclinical platform could be used not only to advance basic scientific knowledge, but we can also use it to facilitate discovery and development tasks within current pharmaceutical pipelines. We are currently pursuing these endeavors with industry and academic collaborators to help us ultimately establish whether the assays and the results obtained with this platform are translatable and predictive of in vivo outcome. So in conclusion, I've shown you today that HSP polymers are new biocompatible materials that can be microfabricated with improved and tunable mechanical properties that match the softest biological tissues. I've also shown you that we can use these materials to make artificial axons, which may be used in the process of developing drugs for uniquely human myelin diseases, and that the properties of the materials that make up these artificial axons can be harnessed to improve the resolution and throughput of production, which can facilitate these applications. So this was an extremely collaborative and interdisciplinary effort, and I want to make sure I acknowledge everyone that was involved, including my research group, my advisors, thesis committee, a lot of really good collaborators at MIT, at the New York Stem Cell Foundation, and Sanofi, and also all the Europes that have played important roles within all the projects I've done during my PhD. So, as well as my funding, of course. So thank you all for listening, and I look forward to our discussion. Thank you.
Thank you very much, Daniela. Of course, you can react to Daniela's talk by choosing to give her a reaction like I will. Thank you so much. So I will open the floor to any questions that you might have. And as you're ruminating about questions you might want to ask, let me ask you a question on my own. So in slide 16, you were showing us an example of one of these proteins wrapping around a pillar. If you can possibly bring that, I think it was slide 16.
Is it, can you see it?
No, it's not that one. It's the one that showed the TEM image
yes.
of a cross section. Yes, yes.
Yes.
And so on the left slide, what is the thickness of those wall membranes that are wrapping around?
Yeah, so each layer is around 10 nanometers thick, and there's a periodicity of loose and compact layers, but it's all within 1 to 10 orders of magnitude thickness, and it varies. Yeah, yes. each layer. But the whole thing, the whole thickness of the sheath is around 1 micron or less.
So the number of layers I would expect would mechanically affect the property. Would change the mechanical Young modulus, right, of the entire structure, or is that the wrong way of thinking of it? I would expect the thicker these layers that you might find a higher Young's modulus.
Yeah, that's interesting. So you mean, so we have the naked artificial axons, so these red,
Yes,
red pillars which have their own mechanical properties, and once they're wrapped in this green membrane, which would correspond to this. So this is just
yes,
a fluorescent image. So now the effective Young's modulus of the entire structure may be different, right?
yes, yes,
Yes, that's maybe true. I am not sure that anyone. It's possible that there's some work out there. I'm not aware of any work on the mechanical properties, specifically elastic modulus of myelin, like how does it change the stiffness of a neuron, for example, and then cells interacting with the neuron, would they be able to sense those difference in mechanical properties? That's a very interesting question, that I don't think we know enough about, but there may be something out there. I'm just not aware of it. But once it's wrapped, generally, once the cell wraps a segment of myelin around the neuron, cells don't really interact directly with that. So some other cell types like the astrocytes will interact with the neuron on the gaps where there is no myelin. So those nodes of Ranvier, for example. And then oligodendrocytes would wrap other areas that are not covered in the sheath already. So it'll be interesting to pursue and see like what relevance it really has.
Um, any other questions? Let me just quickly check a chat. The other thing that, as I'm also checking for other questions, is there an understanding? The choice of the pillar heights that you have chosen, does that in any way affect the interaction with biological material you place on top?
Yeah, that's also interesting. So let me find this slide. The length that we shows, it's long enough that it allows us to capture most of the length of single myelin segments observed in vivo. So you can see here in this length distribution, and this
Mm-hmm,
was
mm-hmm.
done using the horizontal orientation, that
Yes, yes.
cells can wrap segments up to 110 up to 120 microns long, and this distribution is quite representative of what people see in vivo. So it would allow a cell to wrap at least one myelin sheath
Mm-hmm.
per fiber, right? So they could
Yeah,
be
yeah.
made longer and they could be made shorter, and I don't think it's really known whether there's a permissive length. So as a minimum permissive length for an internode to be wrapped. If we make them longer, then it just gives us the opportunity to have more than one internode.
The choice you have made clearly is to apply this knowledge and technology towards biology and towards new medical applications. People like me who study electromechanics or electrical engineering in general might find this mechanical properties of these materials quite unique, in that you can fine tune them down to kilopascal. I believe you said 100 kilopascal was one of the numbers.
Yeah.
Those are extremely soft surfaces, and yet if you can dial them in reproducibly, you might have an ability to generate electromechanical devices that might take a format of a thin sheet with a very low Young's modulus. Is that been explored inside your lab or to your knowledge?
So we haven't had a chance. It's just a couple of people working on this, and me mostly driving this effort. So I haven't been able to branch out. But our collaborators in the FANG group, they're more into photonics and optics, and they can see more the applications outside of biology of this, and that's sort of what pushed us to pursue this. The self-writing of the polymer fibers. And we certainly see that there may be other applications, the coupling of the mechanical properties that we can achieve with this process of self-propagation are very interesting for a wide variety of fields, including things like, solar cells and other things that I don't really know a lot about. But we're always happy to talk to people about other ways in which this could be useful. I feel like we could have a bunch of different projects, just starting with this material.
Excellent. Let me ask you one last question, which is, so this particular process of patterning, I know again, this is related to the details of your process. What is the minimal resolution that is achievable with this UV writing as you have it here? How small of a pillar in diameter could you generate?
Yeah, that's very interesting. We're right now, so I was not very familiar with this, but it's so interesting. We're now doing a lot of literature research and working on our review paper. The goal was to work on an actual project, but because of COVID, we couldn't do any experiments, so we've just been reading up and writing. But the minimum resolution, I'm not exactly sure. So it's just limited by experimental parameters, right? We found that with our material, we could go down to these very thin fibers that are below 1 micron in diameter. So if you could theoretically, if you could the process, you still have enough resin around it, you just let it go. It should keep growing until you deplete or stop the light source or deplete the resin. So I think we can definitely reach the nanometer scale. Then the problem, at least for our application, is how do you collect the fibers in a way that you can still have a uniform enough array to use it for what we want. We don't want a random collection of fibers. But we're definitely exploring, what are the limits of what we can achieve. It's definitely pushes the limit of the technique we were using before, the 3D printing below its theoretical resolution.
I have one very last question. This is from Shardul, who first of all says, great talk, like I agree with him. Following by the next question, which says, the method you're looking for development of hybrid polymers do have defects incorporated in them, or kind of an impurity that can accidentally be introduced. How do you avoid having defects in the process?
Well, so I unfortunately also haven't had the chance, after I developed the polymer, it started working very well, and I just delved into, the application. So which is why we're now going back and looking at, what makes this a special material. So we haven't even done proper characterization, to look at what these networks really look like. But my intuition is that the defects are helping impart the properties that we see in these materials. And because we have these two very different monomers, this hydrophobic short linker, which is also used in, sorry, I'm trying to get to here, in a stoichiometric excess, so this HTDA. And then we have this very large hydrophilic monomer. I think we probably have domains, hydrophobic domains, hydrophilic domains, and all sorts of defects that sort of help impart this biocompatibility, as well as mechanical properties and this optical behavior that we see. So yes, I'm sure there are many defects. We need to understand what they are and what impact they have on the material properties. So this is a whole other project.
Well, you certainly, through your work, have opened up a whole set of ideas.
Yes.
No, Daniela, I very much appreciate you indeed participating today, giving us a fantastic talk.
Thank you
so much. No, I really appreciate it. Thank you so much for joining us. I look forward to the next seminar, which is going to happen this coming Thursday. MIT graduation is this week, so it's a busy week. But if you do have time at 11 o'clock on Thursday, please join us. The talk will be on perovskite quantum dots as potentially scalable quantum light emitters by Mujiba Wendy's group. All right. That being said, again, thank you very, very much for joining us today. Daniela, again, thank you very much for a great talk. And we'll see you hopefully on Thursday. All right.
Yes. Have a
Take
good
care.
day.
You too. Bye-bye.